Good afternoon, everyone, and welcome to Third Thursday for July. This is our first noon hour Third Thursday, and Kate tells me that we had a significantly higher number of registrants for this one than we did for last July when we were starting at 9 o'clock. So I'm guessing that a lot of you have already been working in the vineyard and you're taking a bit of a respite from that. So welcome. Thanks for coming. Super interesting presentation today on reducing the carbon footprint of fermentation and biodiesel from carbon dioxide captured during fermentation. So really interesting in terms of our sustainability efforts. Dr. Leonard Lermer is from Okanagan College and BCBTAC. He's an organic chemistry professor at the college, and he's also been working with the BCBTAC Beverage Research Center for the past 4 years. He's been both teaching organic chemistry and involved in many interdisciplinary research projects for more than 23 years. After receiving his BSc from the University of Lethbridge, Dr. Lermer obtained his PhD in synthetic organic chemistry with Dr. Larry Wheeler at UBC. He then did postdoctoral work in natural products with botanist Dr. Neil Towers. So welcome, Dr. Lermer, and I'll let you take over. I can't hear you. Yeah.
There we go.
Okay,
there we go.
Good.
Thank you. So I'm just sharing my screen. I'll go through the presentation and I'll take questions, I guess, at the end. Which will work with the Zoom format. So thank you for coming to listen to my talk. I feel like I'm presenting as like, what did I do last summer? And this is it. Most of the research I do now is mostly with the wine, beer, and ciders. And this is a little bit of a switch. So the project that I worked on was to collect CO2 from a fermentation system. And the project came from, like most of our projects at the BCBTAC, comes from outside interests. And this one was really brought forward to us by Tin Whistle, which I have on this first slide. It's Alexis and Tim from Tin Whistle. And she's very interested in sustainability efforts and bringing her brewery to be one of the first zero carbon footprint brewery in the area. And also she comes from a background from the aquarium in Vancouver, and that's why she is really sort of passionate about sustainability, which it's more and more people are, which is great. So where this came from, she came talk to me about what they're doing, and when I started looking at it and the amount of CO2 that comes off a fermentation tank, I was actually quite surprised at how significant it was. I'd also like to acknowledge the IAF, the Agriculture Foundation, for funding this support. It was a lot of fun project with a group of students. Okay, needless to say, it doesn't take many. I mean, weekly we see, this was just last week, weekly we see issues with global warming and heat heat issues. And so May was one of the warmest Mays in our area anyways. I wasn't here to enjoy it, but I was in a cooler climate in Paris at the time. So anyways, but we obviously have issues with global warming. I'm just going to quickly go through a couple of global warming slides because it lines up. So here, obviously, when we look at global warming, we have to look at trends over 20 to 30 years, not just, what happened last week, what happened last month. And this is a great thing, looking back historically at the CO2 levels in time, and now how they've been skyrocketing. In 2021, they were saying we're pumping 35 billion tons of CO2 into the environment annually. And at that time, we were only collecting, various processes, 40 million tons. So a pretty small amount. I don't need to talk to anyone involved in grapes understands they're a bellwether agriculture, and how it's affecting grapes is quite significant, how the temperatures change. So this was a slide showing the temperature change with the obviously overlapping with CO2 emissions. And this one ends at 400. We're basically at 450 ppm right now. It's gone up quite a bit in the last couple of years since this slide was made. And this one, I was just in Burgundy, and I thought this was interesting going back to 1857. One of the wineries had recorded their dates of harvest, and obviously this is always the same Pinot from the same fields, and they typically fluctuate between September and October, and now they're obviously producing it more. And one of the interesting things working with the grape industry on this of global warming is there's records going back for hundreds of years on these kind of topics. Typically in Burgundy, they say their date now is 13 days on average earlier than it was in 1989. So pretty significant changes. And they actually have test plots higher up looking at different, an area that really is known for two types of grapes only, 2 varieties, now are looking at other varieties for the future in advance of global warming changes. So anyways, to look at BC, when we were asked to look at collecting CO2, we want to look at the tank. So a typical tank, a 5,000 liter tank, if you calculate based on the percent ethanol, so I took 13.5% because it's right between 15 and 12. So between the typical white and typical red, and roughly calculated using the ethanol, the amount of CO2 that's produced. So on a 5,000 liter tank, you're roughly producing 500 kilograms of CO2, which is about 40 times the volume of the tank because you're going into the gas phase. So it's quite a significant amount that's produced especially with the beer. It's within a 2-day period. For BC, I'm using data that I got a few years ago from the tons and converting it into the amount of wine that was produced. UC Davis has about 80%, and I'm using that to get a ballpark figure of the fact that in BC alone, we produce about 2 million kilograms of CO2. So 2,000 tons of CO2 across BC from the wine fermentation industry. So we wanted to really kind of focus on what can we do with that and how can we trap it and come up with a way of doing it that was efficient. What does 2 billion tons equate to is a good slide. It actually, when you look at the grand scheme of other sustainable efforts that can be done, when we're looking at 600 cars, or I can't see the slide, but a certain number of homes, sadly there it is a significant amount, but it also tends not, there's other larger areas of where reductions can occur. This one is a carbon footprint of the wine. So from the fermentation alone, it produces about 5% of the total input. So there's the phase 1 in the vineyard. There's a breakdown of all the areas of of where CO2 is used for the bottle. So about 1.54 kilograms of CO2 per bottle. From CO2 alone in the fermentation, it's about 0.1, just, so a 5% of this whole total thing. But while going through this process, we were also looking at ways of doing that. And I got to admit, the BC wine industry has really focused on ways of looking at sustainability. And so even, again, recent news about this one's the crush pad, but I know I was just going to talk with a number of people. When I was at Friend and the guys were talking about using lighter bottles, which if you look, bottling is actually a significant cost of CO2 footprint. Anyways, but our focus is mainly on CO2. So I'm an organic chemist. I have to have a slide that has some structures in it. So this is just showing you, the breakdown of fermentation, which I'm sure you're familiar with, and just the fact that for every ethanol molecule, you essentially are producing 1 CO2 molecule, and the molecular weight actually is very close. So basically much for every gram of ethanol you're producing in your wine, you're producing a gram of carbon dioxide. So where we looked at we looked at 3 processes. One was, first we looked at and calculated the amount of CO2 that's being produced in 2 days and kind of realized we can't really bring, because initially the clients wanted us to see if we could put an algae tank on site and have it processed right there. But it just didn't make sense with the amount of CO2 that was produced and in a short period of time. So we looked at other options, and one was to do this carbon capture and store it and then bring it to a site and be able to spend more time dealing with the amount of CO2. The second part is the production. Why we chose this route was in the process we're creating through algae. we are creating oxygen in the process in the bioreactor. And then it also afforded us a way of what do we do with the material at the end? And so that's why we went with the algae system instead of others. So here are another other systems that people are looking at, simply just pumping it into volcanic rock, and the volcanic rock has chemicals that we react with the CO2 to trap it. So it's not just storing it in a vault. Bicarb is a very common one where you're trapping with sodium hydroxide to make bicarb. The real interesting, and succinate is a fairly new one where they're taking CO2 and making succinic acid. The real problem is what do you do with all these things at the end afterwards? CO2 into seawater is great, but you do have issues with changing pH already with CO2 going into the seawater. So we looked at this probably because of the fact that it gives us, one, it allows us to produce oxygen in the process by utilizing more light. And then two, we chose chlorella because it has a huge 50% of it is fat. So we chose something that was going to produce a lot of oil. And obviously it grows very, very, very quickly. Other reasons for using chlorella, it can also be used for human consumption, animal feed. It does have a significant protein content. There's a lot of actual other components in chlorella that are considered beneficial. So there's other reasons for it. Now, our system is actually mostly put together in biosafe conditions. So we could actually create human food out of it, but that wasn't our intent. Okay. We looked at, when we chose this, we also looked at other systems. So this is a system from University of Kentucky. We did a lot of literature searching with my students on what do we want to do and what do we want to create. This one was University of Kentucky. And we see like this one, they're taking CO2 from a coal plant and putting it through these algae systems. And I just didn't see this as a viable option to put on site at a winery or a distillery. Partly because of the space, and partly there's inherent lack of efficiency in the system. The key thing with growing algae quickly is the penetration of light through the algae system. And through even these tubes, you can see there's tubes behind other tubes that are not going to be able to produce enough light. These guys are actually, their system works quite well, but they're looking at putting in a system that takes up 100 acres, which is a large area. There's other very simple systems that just have fields or tanks open, and they use solar to do it. They produce a very low density, and we needed something that was going to be able to handle CO2 at a fairly quicker rate. So that's why we went with the system that we came up with, which we'll get to in a second. So the whole design philosophy, we sat with our students and thought about what it looked like. I'm actually going to flip to the next slide, sort of showing you. We sat at a big board and went through everything, and it's kind of, we're not artists. We're putting down just went through basically a big brainstorming session. And the key thing to our design philosophy was making something that would be adoptable to the industry. There's no point in building something that wouldn't work within the industry. And a good part about with that was, well, one, making it inexpensive, obviously, but something that's going to work, even though it's relatively cheap, but will work and be really useful. We wanted to make it mobile. So you can see we picked actually an IBC tank to design our system in, because they inherently have they can be used by pallet jacks, which I've used because I've worked with wine in some of the wineries, or forklifts. They're easily move aroundable in the industry already. We wanted something that was a relatively small footprint. So if we do have to install anything in a winery or distillery, it would not take up any space and not get in the way of daily operations. That's why the next thing, we wanted it fully automated. We didn't want something that the winemaker or the brewer was going to affect them. They want to focus on making good wine, making good beer. They don't want to be thinking about what do we do here. The next thing obviously is connectivity. We just made sure everything, typically we use tri-clamps and things that we know will be adaptable to what the industry is using. And then scalability, we obviously built a prototype, but I wanted something that we could rapidly increase the volume if we wanted to. All right. So that's the design. We did go talk to, once we had our design kind of came, we did talk to Peter Ralph, who is a professor at Sydney, who's a marine biologist, an expert in algae with marine things. And he's worked on a project kind of similar to this. And it was really nice. The one thing I found about working in what we were working on was anytime you talk to somebody, people were really, really happy to to give you advice and to talk. So he spent 2.5 hours going over and talking to my students on Zoom, which is a really great thing. Is he's on the other side of the planet, and we were able to sit and talk to him about some of the trials and tribulations that he had. We saw inherent flaws in some of his design, although we didn't want to say, well, why did you do this? Because it's wrong. But he was actually very forthright in saying, yeah, we did this because, and we would do it a different way. Anyways, it was really great to have my students sit and talk to somebody from the other side of the world, and move us forward, and also tell us, yes, we're on the really right track in our design. So here's our build. The student that's with me in the picture, her mom's a biology professor, and she's an engineering student at UBC in Vancouver who came back for the summer to work on this project. She's working on environmental engineering. So this project worked really well for her. She was great. One of the best students I've worked with in quite a few years from Summerland. So anyway, so the idea behind it, just going quickly through it, was to build it in ICB totes. We picked the 1250, slightly larger one than the 1000, because it had the right height. So according to the literature, as a scientist, I'm reading papers, we're just saying, if we bubble CO2 at the bottom, by the time it reaches about 3.5 feet, all the CO2 that we bubbled in is actually going to be used, which actually quite surprised me, because I didn't realize that chlorella could absorb it that quickly. But I was, basing the design on what we could read. And then we wanted something that had removable light. So this is the key thing to our design that's different from any other design that I've seen out there, is that we wanted to focus on how do we get in a small area very good light penetration into the system. So because of new LED technology and the lights being so efficient and not producing heat, we basically just put them into the liquid. So most tanks, or most things, the light's penetrating from the outside. So the tank is restrained to being a certain size, because otherwise the chlorella absorbs it before it can penetrate further, and it's just a waste of space. And so in our system, we put all the lights, there's only a foot between each one. So they only have 6 inches of light is needed to penetrate, which is kind of the ideal. And that's the key difference in the system that we came up with, was coming up with something like that. Other than that, we have bubbling stones, which is very common. We're just using air to lift up and cause turbulence in the tank, so it causes turnover. Chlorella is quite a sensitive material, and so organism, and so if you're using a regular pump, it'll actually blow the chlorella apart. So even in the big tanks, they just use kind of big paddle wheels, anything that can move things around without a lot of pressure. And then we just set up a lot of monitoring systems. So that's a sort of a quick picture of the tank, and the key differences that we came up with. We also wanted to be able to manipulate the light. There's still, even now, there's lots of studies going on about what's the best light. Obviously, for photosynthesis, it's mostly taking the blue and the red, which is why we see chlorella as green. We take out the blue and the red and we see the green. And there are a lot of hydroponic lights that have different amounts of blue and green. We do have blue and green in our lights. We also have full spectrum lights as well. And we have the ability very quickly to pull the lights out of these tubes and put other lights in. So we were playing around with lighting, which is a key thing. And like we said, this area, we can't just read off, because it's a biological entity. You can say, oh, blue does this part for photosynthesis, and green and red does this bit. But you can see here, even in this paper, which was only a year ago, they're looking at the effects of the two of them together. And that's why it's it's more complex than just straightforward, this does this. So we wanted the flexibility. So there's the, I don't know why that, okay. So here, the nice thing about it with light and light in the tubes, we had to worry about temperature. The lights from the white light, broad spectrum light, actually are really, really efficient LEDs and really produce very little temperature. We didn't have to worry about it at all. The red and the blue, they're inherently problem with getting just especially blue, you tend to produce more heat. So we needed to dissipate the heat. So they're sitting there wrapped around aluminum pipes, and we have just simple little muffin fans. So one of the few things that we got, you can see they're red in there, they're 3D printed. We got some help with some 3D printers. One of the few things that isn't off the shelf. And that just blows air down the pipe. It comes out around and blows air right out of the pipe. So a very simple system that's efficient, doesn't use a lot of electricity to keep them cool. And we have them at 30 degrees, which is in the range that we want. Anything in that range. We don't want to warm the tank up too much. And actually, as it turns out, we were kind of hoping that it would warm the tank up a little bit, but it didn't at all. Anyway, so very easy way to keep them cool, and allowed us to change the lights out very quickly. So there's the two different systems with the white light, full spectrum light, and the violet is a mixture of the blue and the red. And we can adjust the amount of light that we have for each one to try to maximize our growth rates. And so that's essentially the tank itself, our bioreactor. So we've taken something. This is about, I think about it equates to about eight hectares or 8 acres of forest is put into a 4x4 box for the amount of oxygen that's produced is kind of the rough calculation. Okay. So the next system, actually, we have the bioreactor in hand. We moved to carbon capture. Well, we were doing it conjunction, but we looked to a very common approach already for carbon capture is a balloon system. These systems, they exist in industry, but they're quite expensive. They're only, so a system like this that I'm showing you is for scrubbing CO2. So they take the CO2 off a fermentation tank and they scrub it with carbon filters, and then they reutilize that. So Molson and the big massive Molson plant in Vancouver uses this, but because of their cost prohibitive to any of the, well, certainly a winery and certainly even the microbrewery systems. So, and only the biggest breweries have them because of their cost. So here's our system that does pretty much the same thing at a much reduced cost. So we basically sold our methodology. And also I did talk to one of the guys actually at SellerTech who worked up in the oil and gas industry doing CO2 collection, bouncing ideas off of whether this simple thing would work. And he was impressed with how the whole system, he had a few questions, but it really worked well. So we simply, the one ICB tank that you see there has, basically it's a balloon system. We have a pressure valve in it for release as a safety valve, but the pressure never rose over 1 atmosphere barrel. So it's just the balloon is very pliable that we chose. And so we wanted something where just slight increase in pressure of CO2 in the tanks would actually fill the balloon. And then once the balloon's filled, it goes through a bubbler. It's a foam trap. It goes into a foam trap, then into the balloon. And then from the balloon, we're just used regular compressors at this time to trap the CO2. So that's our whole system right there is the whole system of 3 tanks. It's contained in 3 simple systems. The only thing we have to bring to the winery or the brewery right now is the 2 tanks that hold the compressor and the balloon. So we can easily move it with a forklift. On my Toyota, was able to take both of them. And this is what it looked like in the brewery itself. We just, again, use tri-clamps to put it through. That's our foam trap is the half plastic barrel, rain barrel. And basically it's just a water trap. We actually don't get foam off these large systems, but we kept it in place anyways. It acts as a gas trap, so we never have to worry about anything going back into the system. And it's simply just a water barrel that the bubbles into and then bubbles out. So before we were there, they used to just put it into a bucket and bubble the CO2 through right into the brewery, which surprised me that it wasn't vented outside, but they never had trouble with it. Anyway, so we go in, it fills up the tank. We call that tank, the white tank, the lung, because it basically blows up a balloon. Once it's inflated to a certain volume, it triggers the compressor to compress, and it pumps the CO2 into the tanks. And then once it gets to a small, it shuts the compressor off. So we basically are just sitting there with Matt, who's the brewer, and just chatting for the couple hours that we were collecting CO2. Anyways, the nice thing about this was the very first time we didn't have to do any troubleshooting at all. It worked beautifully. Nothing went wrong whatsoever, which is kind of surprising when you're engineering something for the first time. And then we take that CO2 back to the lab. So this is the lab. Part of it, because we're going to not from lab scale, but actually to a moderately sized scale, we couldn't just use our typical blend that we use for small bottle growing of algae. So it turns out we tried different fertilizers, but this one works the best, which is just an off-the-shelf fertilizer with 10, 52, 10 concentrations. Anyways, they worked really well to throw into the big tank. All our bits and pieces were specifically bought off the shelf. We didn't want to have anything that we had to design or make specifically because we wanted the system to be as cost efficient as possible. So the first tank is just our small system. So when we buy the algae, it comes in a 1 liter bottle, and we pitch it into the medium-sized tank, which is just an aquarium. And that's your very typical, we just put blue and red lights on the outside to grow, and then quickly it turns green. And as we bring up the algae, we can pitch it into the big tank. So it works exactly like typical to yeast. You want to have a certain amount of yeast that goes into your tank. Same thing, we want to have a certain amount of chlorella that goes into the big tank so that it wins out any competition. All right, so here are two. So the person who's pitching the algae into the big tank, I was away at this time, but I asked him to do it for me. He's Luis. He worked at in pulp and paper, so he's actually quite familiar with the yeast strains. So he was a very useful instructor who came and helped us a few times. Luis on the right, he's one of the students who's been working with me and still working with me because we're working at bringing this up to higher efficiencies. And he's was working with sustainable building construction. He's just finished his degree, but his original degree was electronics engineering before he moved to Canada. And he's been a great help on this project. Okay, so quickly we go from a very light blue color, the blue colors from the dye and in the fertilizer, into within a couple of days we're completely green already. This is showing a picture from the light, just showing you how the blue and red light gets absorbed so quickly into the solution. But it worked really well. System worked really well. Our algae growth was very quick, and we haven't even really been changing the lighting systems at all. We've been changing other factors to see if we can increase CO2 production. So we do a bunch of analysis to see the parameters that we're measuring all the time. We do have, specific pH probes. Looking for time. Okay. And various CO2 input outputs. One of the systems that we do use, if you see, is a Senai, which is an off-the-shelf thing for usually used in aquariums or large aquariums, but it was already Wi-Fi enabled, and it was just a very simple system for us to do a lot of analysis. We actually do use this information more than we do the other ones. But you can see in the my device, the lux, if you can see my thing, it's already dropping. It shows how quickly the luminosity of the light is decreasing very quickly over time, which is because the algae is absorbing it. Anyways, we do work with the BTAC, and they allowed us to take samples and do analysis similar to yeast analysis, yeast counts, same system with their microscopes to do counting, and we can see just like we do with yeast counts on a very small scale. We can sit there and we can count them and then come up with an idea of what's going on. It turns out what we do, just like with yeast strain, when we add the yeast, we have a lag, or add the chlorella, we get a lag factor, we get a log factor, and then we get a sustainability of growth. And we're getting that. We're seeing what's happening in our tank is we reach sustainability, and we have chlorella dying at the same rate that they're multiplying, and the chlorella that die tends to drop to the bottom. So we actually don't measure the full density, which is one of the why it's being at a sustained level, even though it's growing quickly. So anyways, this is a slide where we were trying to calculate using the same method as yeast, but we're finding that it's not really giving us real true values for the rate of our growth in the tank. Anyways. And then here, I think this is showing you one of the problems with our tank. You can see something moving around on the slide. That's actually a paramecium that contaminated the large tank. With the smaller tank systems, we were always keeping them enclosed. With this one, because it's the same thing with doing large tank systems, we weren't too worried about it. We were thinking that, we created an environment that was, according to the literature, was supposed to not allow paramecium to grow because their pH was brought up to about 9 with carbonate. And so theory, paramecium will grow. Now it's not so bad because the paramecium will produce biofuel when we do the same process, but it is eating up our chlorella. And so we have the picture, you can see the chlorella inside of a paramecium that's been expanded. So we're trying, it's very simple for us. We just have to decontaminate and then put air filters on the inside, and then an air filter on the outflow of the CO2, so that we don't have to worry about paramecium contamination. Anyways, things that we learned along the way, which we didn't think we'd have a problem with. All right, what else do I think that's about it. So here, this is actually a slide which shows how efficient our system really is. So we're taking CO2, putting it in at a certain concentration. We're bubbling into the tank, and we do actually show with a sensor at the bottom and the top, we do see that we're actually extracting about 70, 80, roughly, typically 80%, 80 to 90%. I think the average, I can't see it, it's hidden behind the slide here, but roughly between 70 to 80% of the CO2 is actually pulled out of the system. So what I have there is, it actually opens up a whole area of, we're collecting CO2 from a point source, but because we're able to extract these numbers here, we're actually not even we haven't even started putting in the CO2 from the fermenter. We were just running air through it, and we were extracting the CO2 straight out of atmospheric air. So at 450 ppm, going way down. So it also opens up the whole area of just direct air capture, where we can just simply run the system, pull CO2 out of the atmosphere. And so someone from a much further area is like, okay, well, we're producing this CO2, and that's where the whole idea, I understand much better the whole idea of your carbon, sharing. It's like, okay, we're producing carbon here, but you're taking it in here. We'll pay for those credits. And I was like, it makes sense when you have systems like this. It really also illustrates that CO2 and CO2 issues are really a global thing. It doesn't matter what we're doing unless other people in the rest of the world are also doing it too, because it affects our total environment of our environment. Okay. Speaking of which, well, after putting this together and doing the math, I got on a plane and went to Paris, and the guy, our pilot, talked about how much we had to wait for a bit while they were filling the fuel, and talked about how much fuel went in. I just went, oh, it's kind of depressing for our global warming efforts. But basically, I didn't fly an Airbus, but a similar plane, and the Boeing plane, but basically the Airbus, if it's going back and forth between here, Calgary and Paris 5 times, it equals to all the CO2 from the fermentation tank, not from all the issues with the BC wine industry, but from the fermentation CO2 itself. So there's obviously other ways that, things have to be are looked at for the sustainability of our environment. And again, we could sit there and run, like I said, our one simple 1,200 liter 4x4x4 tank can equate to about 8 hectares of land. And I can't stress, obviously, from the Okanagan, land is of short supply. I'm not going to take a field and put 100 acres and convert that into algae system. That's not feasible, but we can certainly have a warehouse with lots of tanks in it, and basically have 100 acres of forest in a small warehouse space. Okay, so moving on, this is what the algae looks like when we ferment it. We're obviously doing it on a lab scale, or we're simply just using a filter to collect it. And the key issue with algae and any algae system is dewatering. You can see in the ponds, it was like 100-fold less algae per liter than what we're producing. But we still have problems. We still have problems with dewatering. It's a difficult, and the reason why it's difficult is because the algae will, if you try to do a simple filtration, the algae will bind and block the filters fairly quickly. There is a company in California that says that they've sort of solved this problem. And the reason why they're trying to do it is filtration is the easiest way and the cheapest way to do it. Typically, algae is done, they first often use chemicals to cause it to fluctuate and then settle, and then they filter it. And also they use a flow-through centrifuge. And using a centrifuge, kind of counterintuitive, because it's a huge energy cost to use a centrifuge to isolate the algae. So when you go to buy your spirulina at the drugstore, most of the cost for that algae in the drugstore is because of drying into a powder, and that's because of the cost of the centrifuge. So one of the cool ways that's come up, which we don't have yet or hoping to get eventually, is electrocoagulation. This was actually developed for wastewater treatment at one point, and turns out the algae itself has a slight negative charge on it, and that's what keeps them from fluctuating together in solution. And by just putting a small charge on the cathode at the bottom of the plate, it causes the algae to rise to the top of the tank, and then they just have a paddle wheel that takes off the algae. So it's a beautiful, elegant, thought out system that works quite well. So we focus primarily on the tank itself and the collection system itself. And these are downstream systems that other people are working on that hopefully we can get the equipment to utilize, which is where we're looking at trying to get some more funding for. Okay. Taking the algae and making biofuel from it is fairly straightforward. It's a simple transesterification process. There have been people like the picture on the right is soybean oil that was converted, simply just adding methoxide, which is just made from methanol and sodium hydroxide, and getting it to react to produce the glycerol and the methyl esters. So biofuel itself is just methylated esters of the fat. The small tube that I have was actually made from the algae itself, just a sample tube that we used. Turns out for us, the transesterification worked better with using acid catalyst instead of base catalyst. So use a little bit of HCl. Key was drying it before, making it a dry solution before, otherwise you tend to produce too much soap which is just the negative charged acid, versus making the methyl ester. So there's some tricks to it chemically, but pretty straightforward. This part was actually the part that's in my wheelhouse was one of the easiest parts to play with. Okay, now where we would like to go with this, where I'm looking for funding, the simple, very quick one is right now we're collecting only about 4 kilograms of CO2 out of the tank into those two 30-gallon tanks, so 60-gallon tanks, because we're using a standard compressor that goes to 135. We'd like to work with just a very simple, this is just a scuba tank, which are only scuba tank compressor, which is actually a reasonable one. It's only $2,500. And then there's also already pre-made dewatering systems. We just like to purchase the equipment. Again, to keep it mobile would also be these cage systems for high pressure tanks. Tanks are about 500 a piece, but they come already pre-made in these systems that are designed to pick up. So it's a very safe way of moving a large amount of highly compressed gases. Versus the really big tanks, which you really don't want to move. But the nice thing, the reason why we want to do this is so we can leave the tank, we can bring a tank and leave the compressor in, but now I can just come pick up the tanks, take them, grow algae with it. Or the other sort of side thing that we'd like to do with it and purchase would be this is an industrial CO2 scrubber, which they're $300,000-$400,000 a piece. We'd like to just get a nice small system so that not only could we grow algae, but we could also reduce the carbon footprint of moving CO2 around into the valley, because right now CO2 has driven in from quite a ways away where they produce it to make bio-grade food grade CO2. And so we'd like to just have a system. It doesn't make sense for one small winery or one small brewery to do it, but if we had one site, we could handle all the CO2 very easily for the whole valley by simply just bringing the CO2 in, cleaning up through these strippers. Basically, they're carbon filters that have a flush system of steam so that they can clean off the CO2 or clean off the carbon once it's trapped the organics, and then basically bring back the CO2 that can be used either they use a lot of it in the breweries more than the wineries, but you certainly use it in the winery too. The other sort of way is, I guess, while we're sitting here and thinking about other things that we can do, is we've looked at some waste products from certainly the brewery, the spent grain, but also the pumice, putting it through a screw press and dewatering. It's a very efficient, inexpensive way of dewatering, brings it down to a certain percentage, and then you'd have to do more. But from a screw press, you actually have it to a system where we could easily make pellets from it and throw it into, this is an electric generator that burns pellets. It's from a Quebec company, But the reason why we're thinking about this is one of the things that we're doing in our system is using light, and we'd like to come up with a way of creating the electricity to run the light systems. Because yes, if you're in Phoenix, you can have it out in the field and grow light, but certainly when you're producing wines, even in September, our amount of daylight is already being reduced. So either by creating using alternative electric source, certainly by using waste material to make that source, it would be a really useful. This is how we're thinking of future endeavors. I've looked at biogas generation as well. There's a company that I have a friend out here who has a potato. I'm in Alberta right now. He has a big, huge potato farm. He's got his own biogas generator system on it, which is quite a cool system to use. But I've looked at, there's a company that's planning on putting a biogas generator at the top of Brenda Mines, which doesn't quite make sense because you have to transport it all the way up the hill. But I've talked to them about including in their design, because when you are going to create a bio waste generator, you have to know what you're going to put into it at the beginning. But I've tried to include, like, in the design, can you put the pumice from a winery into it, and can you put spent grain from all the breweries into it? I've talked to them a little bit, but they've disappeared for the last little while. So that's why I stopped even looking at biogas. But anyways, lots of things to consider in the future. But like I said, going back to what we're doing, we're looking at one small, like the phase 2 reduction of CO2, which is actually, when I look at, after the fact, when I looked at the whole footprint for the winery, it's actually a small piece, but it tags into just overall reduction, but it also tags into how we can do it, not just for the winery, but we can use that to continually produce algae and biofuel and remove CO2 from the atmosphere all year round, not just during harvest. And then also looking at, it's opened up our conversations with looking with the TAC and with other people about how we can look at other ways of helping to reduce the carbon footprint on a number of these aspects to help make the industry more sustainable. And I'd like to thank quite a few of these people. Like I said, it was a really fun project, because anytime I talk to somebody, I say I'm working with students on this bioalgae reactor, because we're buying bits and pieces from people, and they're looking at us kind of strange when we walk in to get them. And they were really happy. And you find people with incredible backgrounds in hydroponics and the better than nature guys, in lighting and LEDs. I also like to thank Logan, who's the chair of the mechanical department. He helped me out quite a bit with the 3D printing of the few things we have, as well as Owen. We had one part that we had to fit the glass into, and he did all the mechanical work on it in the lab, and made an incredibly tight fit, which was beautiful. Anyways, but also just on the local industry, Solar Tech guys, the new line, they really helped us out and sat with me and talked with me about some of this stuff for an hour, a couple hours. So it was really good. And then here's the team that we had, the 3 students. So Jacqueline, as a local student that I talked about, she's at UBC Vancouver in engineering. Edward is an electrical engineering student at the college who's now moved up to UBC to do his 3rd year. And Omar just finished graduating from the sustainability construction. And I just like to again thank you for coming to the talk and listening to me talk about, because I feel like I'm a kid in school talking about what did I do last summer. This was really done mostly last summer. We've continued monitoring mostly with Omar on the methodology, or on the algae, increasing the algae tech. And lastly, I'd like to again thank Investment Ag Foundation for supporting us on this endeavor. Thank you.
Thank you, Dr. Lermer. That was super interesting. I'm going to open up to questions, and hopefully I can ask my question first. Maybe I missed it in the presentation, but did you address the amount of CO2 that's captured by the vines per season?
No, that
right, because
by the vines themselves, no, we didn't.
Because we were looking where the CO2 is coming from, right?
What's that?
That's where the CO2 is coming from, that is released during fermentation.
Yeah.
Right. So
yeah, so the vines themselves are a source of, they're pulling CO2 out of the atmosphere themselves by solar. They're doing photosynthesis. And so they're actually, so what we're doing is taking as the grapes get processed, and they're essentially releasing the CO2 that was bound by solar energy. So the
Yeah.
grapes themselves are a solar energy trap.
So
And what we're doing is essentially recycling it by taking the
yeah.
CO2 and then recreating it back into an organic material that's going to get used again. And yeah, even with biofuel, we are going to re-release that CO2 in the atmosphere. But again, it's CO2, we're reutilizing it multiple times.
Yeah.
And each time we're actually absorbing more solar light if we use solar light to create the algae.
Yeah.
But we're essentially just putting back the CO2 that we've taken out. So it's really what it is as a system is to make biofuel that can be used instead of diesel fuel from the ground, so that it becomes a net zero amount of CO2 in the atmosphere. So
yeah, I'm sure that calculation has been done.
Yeah.
I don't know, maybe Ruth knows more about that, but because that is an important part of the conversation, like for a winery that's trying to achieve net zero, I think, right?
yeah,
Like,
I'm curious. I didn't see actually, because the slide that I had that had the whole economic assessment of CO2 footprint. I don't know if I didn't look at it accurately enough to see if there was a section for CO2 being pulled out of the atmosphere.
There's a vineyard section, but that I'm assuming that CO2 being produced by tractors, etc.
By tractors, yeah. They didn't, I don't think they accounted for they didn't do the subtraction of the CO2 you're pulling out of the atmosphere.
And it's not one-to-one because we're only fermenting the fruit, right? So there's a whole canopy that represents.
But I guess when they're looking at the fruit that you're producing ends up producing CO2 in fermentation. And so it's being released anyways. And then any of the canopy, it's hitting, well, I guess it becomes organic waste.
It depends on how
what happens with it.
Degraded after. Yeah, it's complex. It's a really complex picture, right?
Well, that's where I'd like to use any of the organic material that's left over, like the pumice and like the spent grain, and extract the remaining energy out of it, because that's essentially sitting there as a waste material that's actually been energy created by solar. I mean, really what plants are is a solar battery. It's a battery that's stored energy from solar light.
Well, and because it's it has an annual life cycle, right? It's it's not like trees, right? So that material goes somewhere every year, right? So Ruth, do you have a question?
I do, Kathy. That was super interesting. Thank you for that presentation. I did visit BCBTAC and got to see it happening in person. I took a little video of the, fish tank going and thought it was so cool. But I was just wondering, you were talking about a bit about like carbon calculation. Have you had access to a carbon calculator at all?
No, I haven't done the full carbon economic assessment of the system. We're trying to bring it up to where we know exactly how much carbon we're actually sequestering per day. And once we have that in hand, then we can actually we're actually going to evaluate it in more detail. Okay. But I mean, I have access to online calculators. They don't fit our system perfectly. Yeah, but we can certainly do the calculations ourselves. And once we have a system up, and I think I'm going to have to look at doing a calculation based on, for us, the best way to do it is the amount of CO2 we're putting in versus the amount of CO2 that's going out. Mm-hmm. And that'll tell us a better example of what we're trapping. So I
don't know everything there is to know about carbon calculation, of course, but we are going through the process of building one with all three scopes, scope 1, 2, and 3 of the UN SDGs. And if and when that happens, I'll reach out and see if there's a way that we can make it work for you guys somehow. I don't know exactly how to integrate it, but it seems like it could be possible.
Yeah, no, that'd be great. I'd be happy to work with you guys. And also, like, for us, we're in the, the funding on the project. You can see how we actually did quite a bit with the initial funding that we got. But now it's opened up to, we're looking for more funding to take this to the next level where it actually becomes a really viable thing. Being able to collect 100% of the CO2 and not just a portion of it for a trial project, but to actually take it from this prototype stage to something that could actually be really, I don't think it's going to take a lot more money, but dewatering system would be the biggest one. So,
And have you looked at the sustainable cap funding?
yeah, we're looking at everything right now.
Yeah.
But any suggestion would be great. So we're looking at more. I in the BTAC, I've got so many other projects going on as well at the same time. So sometimes I lose focus on this one, but it keeps coming back to me because it's such an interesting project. I think that has a lot of potential value.
Yeah, I think it fits really neatly into the sustainable cap funding. One of those streams definitely has your name on it. And the other question I had was about what it would cost to install. I know this is a prototype, but what kind of price range are you looking at for installing in a winery?
Well, so the ideas of the tanks, really for the winery and the distillery, what we're looking at, really all you would have would be the compressor on site and not bother to move that. And there are about 3,000, I think, would be enough for the compressor. And then the water, there's a dewatering system for the gas before going into those tanks. And then the tanks themselves are 500 a piece. Depends on how many tanks you need, but really the tanks, I think, would be more in terms of a business model, would probably be more of either a rental where you don't own the tanks, just like a propane system. The tanks just get brought in and brought back out. So the only really thing, would be, from my sort of way I calculate it, would be, under $5,000. And then the tanks would come in, they'd fill them, and we'd take them away, and then bring back like a clean tank of CO2 in the future. Mm-hmm. So I'm looking at trying to keep the price down, as to me, like, it's going to work, but I'll keep a price down where it's not asking for people to put out. Certainly, a lot of the microbreweries have less money than some of the wineries. They're running very tight margins. So something that's very, very obtainable, and I think would be useful. Where, we did a quick calculation on what the state cost savings would be for their CO2 production. And so just with basically bringing back your CO2 at a much reduced price, would actually affect make it economically worthwhile to have it.
Mm-hmm. And I might have missed it. I was a few minutes late here, but did you mention that you were working with any wineries or?
No, well, in other projects we are, but this project was primarily Tin Whistle, the brewery. And then also Okanagan Spirits was also a little bit involved in it, but primarily Tin Whistle was involved. We've always had, because we're so connected with the wine industry is the major industry in the valley. We've always had in our head in the design is something that would work in a winery. Because then the idea behind the mobility of it is because a winery only fermenting for a few months, weeks to months. Some have grapes coming in continuously, but so the smaller wineries, which is we were focusing on too, is if they're only producing for a few months, we just want to bring it in, collect the CO2, and take it away. So there'd be no footprint essentially remaining. You could actually leave it outside and just have the line go outside. We also, the beer tanks already have a line that comes off the top. Some wine tanks do, but a lot of the smaller ones just have a lid. But you could easily adapt a system that, because it's happening at less than at 1 atmosphere, any increased pressure, just a lid with a bit of weight on it and one pipe coming off would work. So we could easily adapt to a fermentation tank in a winery. So we've always had in our head to keep the system what would be useful in the wine industry. And again, winemakers want to make wine and focus on wine. The system's fully automated. The compression system's fully automated, so they don't have to, except for taking and putting the pipe on. That would be the only thing they'd have to do. Or they wouldn't even have to do it because we would do it when we show up.
Yeah,
So-
I worked at a winery where we created sort of a passive collection system. So we closed the tops of the fermenters and then just used that CO2 to blanket like a neighboring tank that was cold settling or finished fermenting or something like that, where
Right.
you're using dry ice or something instead, right?
Yeah.
So, and that was just PVC piping pretty much, like a fitting for the lid and piping. So yeah,
Yeah.
that
Yeah.
part would be a no-brainer for sure.
We just use straight, the typical, 2-inch pipe, regular, the vinyl pipe that typically are used in
Right.
pumping wine across, right?
Right.
So we got that from New Line, because we wanted to use systems where, you know what, you've got the pipe right there, we don't have to bring it. Anyways, I think it's very adaptable to both industries.
Yeah, it would be, yeah. I mean, I wonder if there would be any challenges just adapting it to winery versus brewery, I don't
No,
know.
we've kept it in this, the intent from the beginning of the system was to work in both. And
Right,
again, I worked at a winery briefly where we basically open the door and have to sit there while all the CO2 comes flowing out in the morning before we walk into the building. And I was like, yeah. So it's kind of like, oh, this would be very easy to not have to do that, save you that time of waiting before you're basically not going to worry about CO2 passing out from the CO2 contamination.
yeah. Leonard, you need to look in the chat because you've
Oh.
got a funding offer right there, or contact. So that's a great development. Maybe that means we'll have you back for further developments in a few months, right?
I do have one
Okay.
one more thought that I wanted to just throw out. The when I worked at Sandhill here in Kelowna, they were trialing a setup by Barrel Wise, and I'm not sure if you've heard of them. I think
They're from
UBC.
what's that?
They're from UBC. They were in
Oh.
an engineering.
Okay, there's a group that was doing, were they just doing CO2 capture?
No, actually not at all.
Okay.
But I'm just wondering if their rig, their whole setup would somehow help with facilitating your setup in the winery. So barrel wise, it's about working your barrels easier. So you have a little cart that you drag around and you have like basically a fanny pack with their gizmo that goes into the bung. They have specific bungs and it goes into the bung, and you can do your additions, you can measure
Oh, yeah.
things, all that, like just without actually opening and pouring and whatever into the barrel. So it makes barrel working easier. But I wonder if they're like what they've built would somehow maybe it could be like an add-on for
Possible if it's like if you're doing your ferment in the barrel. We were looking at fermenting in tank primarily because it's both done in the wineries and the breweries and the distilleries too.
Yeah.
So we were looking at fermenting in tank and also as larger volumes, but certainly, the system itself is very flexible. As long as you can get a pipe into it and bring the CO2 will push itself through with anything over 1 atmosphere.
Yeah.
We could easily collect it from barrels. It just means more plumbing.
Yeah. No, I wasn't imagining that you'd be trying to capture from barrels because that sounds like a big mess.
Yeah, it could be done, but
Yeah.
But I wonder if what they've created would somehow help you implement the CO2 capture from tank.
oh,
Anyway.
maybe I'll look into them. So I can look more into them and see if there's some synergistic ways we can put together.
And if you do, they were all about trialing things and it's been a few years now, but I think they'd still be open to it. And they're also a very big producer. So that would be a good way to take a chunk out of CO2 emissions.
Dr. Lermer, you can get in touch with Ruth through SWBC.
Okay.
So if you have further questions, and Kate at any time would be able to hook people up through the Wine Grape Council. So thank you so
Sure,
much
sure.
for this presentation. It was
Yeah,
super interesting, and I'm really happy to have the
It-
recording on our website. And
Okay.
hopefully we'll have you back for more.
Yeah, I'd be happy to. I'm sure that you were asking about the TAC. I'm sure maybe Sonia can do a microbiology talk. She's quite in.
Yeah, maybe for August. Yeah,
Yeah,
so
okay.
the August session will also be a noon to 1 session. The topic is to be announced. If anybody has suggestions for desired topics, let us know. And we'll see you then. We'll see you August 18th, I believe it is. 17th, August 17th. Thanks again.
Thank you.
Bye-bye.